Dr. Sheftall, we're delighted to have you back with us. Ladies and gentlemen, Dr. Sheftall is a surgeon, a plastic surgeon, heart surgeon, medical doctor, one that has served in major capacities in relationship to authorship and professor. So I'm thankful that he's with us today. So Dr. Sheftall, take a moment and tell us about yourself. Thumbnail introduction for those that have not heard you this week. Let them know that they're not listening to a conspiracy theorist, but in reality of what you know. Give us a little insight.
Thank you very much. Now, before I do that, could I please thank Jim Crendon again for putting us together.
Crendon.
Yes, who knows my sister, and that's how that happened. And also to thank you, Pastor Youngblood. You've been a great host and asked great questions, and I've really enjoyed getting to know you this week. So thank you very much.
It's been our blessing. I'm thankful for it.
Okay, now my name's Dr. Sheftall, and I got an undergraduate degree from MIT in physics. Then I went to medical school, and I've done 3 surgery residencies. So I've been well educated. Luckily, been very fortunate to get all that education, and it all came together with COVID. And as you may recall from our last meeting together on Wednesday, I was getting ready to ask you a very interesting question, and that was, can you get the Bible out of To Kill a Mockingbird? You mentioned that you had read it when you were younger, To Kill a Mockingbird, and of course you've almost memorized the Bible, I'm sure.
That's right.
But you would say, no, you wouldn't be able to get the Bible out of To Kill a Mockingbird, but in next generation sequencing, you would be able to do that, because it's a license to lie, if you will. And let me explain what I mean by that. I was in the process, when we ran out of time, of proving that the SARS-2 virus did not exist. And the way that I proved it was by debunking the 3 things that virologists use to demonstrate the existence of the virus. The first one was photographic evidence, which I debunked because you can't tell the difference between an exosome and a viral particle, and both of them appear when cells break down. Or excuse me, some round bodies appear when cells break down, and the virologists were claiming they were viral particles, and of course we have proved now that they are just exosomes. And that's even supported by the Journal of Virology, who says that you can't tell the difference morphologically between an exosome and a viral particle. The second way that they demonstrate the existence of viruses is by showing cytopathic effects supposedly caused by viruses. But of course we did the control experiment, which the virologists have never done, since this technique was developed by Dr. Enders, who received the Nobel Prize for it in 1954. The control experiment, of course, is to run the same process, the same methodology, and the same steps, but don't add the human specimen. Therefore, viruses could not possibly be causing the cytopathic effect. So when you do that, you get the same picture, the same result as you do when you add the human specimen. So that shows you that there's something other than the viruses, other than the human specimen, that's causing the cytopathic effects. And of course, it's the poisonous antibiotics and antimycotics that are added to the mix, and the starvation, which is one of the steps that occurs in the development of cytopathic effects. So we've now debunked 2 things. Now I'm moving on to debunk the third thing that virologists use to demonstrate the existence of viruses, and that is sequencing the viral genome. And the reason that is debunked is because you cannot isolate and purify the virus, okay, or the viral RNA from the bronchoalveolar lavage specimen or nasal swab. You cannot isolate and purify it because the way that that's done is with an ultracentrifuge that separates things by density. And of course, RNA from any source, be it human, bacterial, viral, fungal, will have the same density because it's made up of the same nucleotides. You can separate DNA from RNA because one is deoxyribonucleic acid and the other is just ribonucleic acid. So they're different densities. They can be separated. And of course, you can separate the proteins that you find in the mix from those 2. So because you can't separate the RNA, the viral RNA from the other kinds of RNA, let alone SARS-2 viral RNA from other viral RNA, you can't get a you can't be assured that what you end up with in the genome is only from SARS-2. Do you follow me on
Following
that?
you.
Okay. So let's get now quickly.
In fact,
to
to clarify that for our
Yes.
listeners, you're talking about if you've got a common flu or cold, you're talking about basically the same rudimentary virus.
No, the cold viruses traditionally were caused by coronaviruses, adenoviruses, or rhinoviruses.
Correct.
Whereas the flu has its own separate virus,
Correct.
the influenza virus.
Correct.
Okay, so those are different.
Right.
But just getting back to the deception that's inherent in the next generation sequencing techniques, the way it works is when you have a collection of RNA that you've gotten from the ultracentrifuge, there are little pieces of RNA in it. Like I said, we don't know if they're human or bacterial or fungal or viral. And in the case of the 41-year-old Chinese man that Dr. Fan Wu of China used to sequence, he was one of the 14 people from the Hunan seafood wet market, if you remember. You remember the wet market theory.
Yeah.
Okay. Dr. Fan Wu got 56 million reads out of there. And a read is just a short segment of RNA. Okay, again, we don't know where it came from. It could come from a human. As a matter of fact, in someone's bronchoalveolar lavage fluid, about 95% of it is their own human RNA. Okay. And those 56 million reads get converted to their complementary DNA counterpart. Okay? And that's done because the PCR technique, which we will talk about a lot today,
Yes.
uses DNA, not RNA. That's the only reason for flipping it over to DNA. Now, the way that the genome for the supposed virus is created is those 56 million reads get cut up into small pieces and then reassembled based on bioinformatics algorithms and templates of viruses that have come before that unfortunately were also determined by this fraudulent method. They get reassembled into the genome of the virus you're trying to sequence. Okay? Now, let's pretend, as an analogy, let's look at the To Kill a Mockingbird. Let's say that you've got To Kill a Mockingbird and you want to find the Bible in there. Well, you can't find it in there. However, if you cut the To Kill a Mockingbird up into individual letters, you could rearrange the letters into the Bible. Now, you might come up a little short on some letters, like the word kingdom is probably in the Bible quite a bit, so there may not be enough K's in the book To Kill a Mockingbird. But you can use the PCR technique to amplify the K's. That's how fraudulent it is. You can amplify anything you need to put into whatever genome you want. Okay?
Okay.
You see how that would be done? How you could find the Bible in there if you cut it up into small enough pieces. You just rearrange the order of the letters and you've got the Bible.
So that's a good transition to the PCR testing.
Yes, it is, because I'm going to tell you in a minute how the PCR test works and how it was used to deceive everyone, which is really, people need to know that.
Yes.
Because a lot of people kind of understand a little bit about the PCR test. They know that it's a way to multiply something. They're talking about 1 trillion copies, but they don't know how the copies were made. So just everything is confusing to them. But I'm going to tell you in very simple terms today how the PCR test works. And people will appreciate it, because then they'll really understand how this deception was done.
And for the edification of those that are listening, when we say PCR test,
Yes.
we're talking about the test that was universally used to determine if you have COVID.
That's absolutely right. It's called the RT-PCR, which stands for reverse transcriptase polymerase chain reaction. And the reverse transcriptase part is in there because SARS-CoV-2 is ostensibly an RNA virus. There are RNA viruses and DNA viruses. It's an RNA virus, and it has to be converted over to DNA to work in the polymerase chain reaction amplification method. Okay? Remember, the PCR is a way of amplifying or multiplying a specific sequence. And why don't I go right on to that now and explain how the PCR test works. All right. Okay. There are several steps in the PCR test. It's a method to amplify DNA or RNA so you can have enough material, DNA or RNA, to be able to perform chemical tests on it, such as, is it there? You know, you want to find out, is it there? Such as if there's SARS-2 virus genome in your body, that means at some point you were infected with the SARS-2 virus. Okay?
Correct.
But I'm going to show you how it was all a big deception. Okay? Because think about what the situation that you're in. I have now proved that through showing that next generation sequencing is a fraud, and the other two things, that there was no such thing as the SARS-2 virus. It does not exist.
Digress for just one moment.
Okay.
And we covered that. Some people listening today may not know what you said. Give us a little thumbnail window before we go further with PCR test.
The SARS-2 virus, and therefore the disease COVID-19, did not exist.
Okay.
COVID-19 was a rebranding of other more common diseases, such as what we call the flu.
Okay.
And other things. You know, there was even a motor vehicle accident that was attributed to COVID because someone had a positive test, and so to beef up the statistics, they called that a COVID death. But now that you've got a goal of creating a pandemic disease for the reasons that we mentioned a couple days ago, meaning they didn't want Trump to get reelected, they had a crisis in the repo markets, the cash markets. They didn't want people to assemble because the people were getting very agitated with their governments, as they were, say, the Chinese government down in Hong Kong. And the Chinese government had to send the military in to stop them. They were so effective at blocking the roads and everything. So you've got a goal of creating a pandemic disease. That's if you're one of the evil organizers now. But you don't have a virus or a disease. So how are you going to do it? Well, this is when you enter the fake genome and diagnostic tests that are based on it that can yield a positive result whenever you want. And I'm going to show you how the PCR test was used to yield a positive result when there wasn't even a virus, which is really hard to believe. But I'm going to explain how they did it. The first thing I want to do is explain how PCR works. Okay. And then once people have an idea of that, then they're going to be able to see how the deception was done. But without knowing how the PCR works, it's very hard to understand the deception. So let's talk about how the PCR works. There's steps involved. The first one is sample collection. You know, you've got to get some bronchoalveolar lavage fluid, or swab the nose, or stick the long Q-tip into the nasopharynx and swab in there to get some DNA material that you're trying to determine is SARS-2 or not. Okay?
Okay.
All right. Then you have to extract the DNA from that sample, from the mucus that you got out of someone's nose. Okay. Then here's where the real deception occurs. It occurs in primer design. The primer is a short segment of DNA designed to bind to specific sequences of the DNA extracted from the sample. Okay? Because the DNA, the genome, is going to have certain sequences in it that are like fingerprints on people, that are only unique to that piece of DNA from SARS-2. Okay? So if you can design a primer that is the complement of that piece of the genome, then you can determine when it anneals together, and I'll talk about the steps in a second, whether or not you have SARS-2 in there. If it's a tiny speck, you can amplify it up to 1 trillion times so you can perform the chemical test to determine if it's in there. You follow me?
Follow you.
Okay. Now, so here's where the deception occurs. Again, here's the thing. The primer must be the complement of something that's unique in the genome of SARS-2.
Gotcha.
Okay. And just like fingerprints. Okay. Again, I'm telling you how PCR works. I'll tell you how they deceived you later in just a second.
Okay.
Okay. The next step is amplification. because again, the PCR process is a way to amplify a specific sequence so you get enough of it to perform a chemical test on it, such as, is it there or not? Okay. The next step is amplification. And this occurs during multiple cycles of heating and cooling. Okay? When you heat DNA, it will come apart. Everybody remembers that DNA is a double helix, right? It's two strands that are joined together by covalent bonds, and we know that adenine goes with thymine and guanine goes with cytosine. Everybody remembers that that's been in medical school or studied biology. And when you heat it up, remember, DNA is like a twisted ladder.
That's right.
So the rungs of the ladder will actually come apart at the covalent bonds, okay? And you'll end up with two complementary strands when you heat it very hot. It'll just come apart, okay? And that is when the PCR primer scoots in there to anneal. When you cool it down, it'll anneal to one of the sides of the DNA. And then you also throw in a bunch of loose nucleotides. And the polymerase, which is an enzyme that makes a polymer of nucleotides, will make a copy of what the primer annealed to. And that's how you double the amount at each step.
Yes.
And you may have heard that each step of each cycle in the process doubles the amount of primer or genetic material, okay, that it's finding on the DNA of the SARS-2 virus, supposedly.
Correct.
Okay? So when you think about this, you take something that's 2 to the first power is 2. When you double that, it's 4. Double that, it's 8. And then 16, 32, 64, 128, 256, 512, and 1014. That's 10 doublings. Okay? So if you double it 10 more times, so you get 1,000 after 10 doublings. If you double those 10 more times, you're going to have 1,000,000.
Yes.
And if you double those 10 more times, you'll have 1,000,000,000. And if you double those 10 more times, you'll have 1,000,000,000,000. So that means after 40 doublings, you'll have 1,000,000,000,000 copies of what you started with. Okay, what you were looking for.
Yes.
With the primer on the SARS-2 genome. Okay? All right. Now that's important to know. Now, the last step that I just explained, when you put in the loose nucleotides so you have the material to make the copies, is called extension. All right. Now, here's the problem, and here's where a great analogy comes in, which you'll enjoy this. After you've heated and cooled the soup to extreme temperatures many times, the enzymes start to wear out. The polymerase will wear out and start making mistakes. Okay? And of course, these are copying errors because it's taking that little piece of DNA that the primer attaches to, and it's copying the primer with the loose nucleotides. But it makes a mistake. It puts in a G instead of an A, so a C binds to it instead of a T. You follow me? It makes little mistakes. And the problem is that those mistakes that are made along the way to get to 1,000,000,000,000 copies are propagated through the system after the mistake is made. And the analogy is, do you remember in maybe second grade, the teacher played a game called telephone with everybody? Mm-hmm. And they would set everybody in a circle and give a simple sentence to the child in the first seat. Maybe it's a little girl, and you'd say, Dr. Youngblood is a great preacher. Okay, you would tell her that. And then her job was to tell the next child in line, in the circle, right, Dr. Youngblood is a great preacher. And each child is supposed to repeat that all the way around to the end of the circle. But of course, when you get to the end of the circle, if you remember playing this, it could be something about a purple school bus.
Yes.
It could be completely different.
That's right.
Because the kids make mistakes when they repeat it. And once you make a mistake, that mistake gets propagated through until another mistake is made on top of it. And by the you get to the end, it's completely different.
Well, I've used the illustration. It's like the old thermal copier. You can put the original in there, and after you take the thermal copy, then take that copy and make a copy of it, and that copy and make a copy of it. After about 10 copies, it's so watered down you can't read it.
Exactly. That's a very good analogy, except that when you make a sentence that you're repeating from person to person, it would be a corpuscle or a package error.
Yes.
It wouldn't be a partial error like the Xerox copying would
Right.
be. It would just get fuzzier and fuzzier,
Yeah.
and you wouldn't be able to read it. But this, they have to make a sentence that they get to the next kid in line. So the sentence will never go from something about a purple school bus back to Dr. Youngblood being a good preacher. That would be a violation of the second law of thermodynamics.
Yes.
Okay, so the same thing happens with PCR. Once a mistake is made in the copying, that mistake gets copied every time until it again gets a mistake done to it. Yes. So that's a very important concept to understand, because if you go to 40 cycles and the enzymes get worn out and they start making a lot of mistakes, you can find anything in the original mix. Okay? Think of it if it was just four base pairs long. Say it was AAAG. And after 25 copyings, these worn out enzymes are going to make it AGGG by having made a mistake. And then 10 more copyings, it's going to be CCCA. You know, it's just completely different. So eventually, you can see how you would find just about any combination of four base pairs there if you run the copying long enough. And the analogy with the kids in the school playing telephone is, if you had a circle that was 400 kids long, can you imagine how big of a mistake it would make by the end? So that's the idea. And that's one of the ways that the deception occurs. And I'll explain why in just a second. But just to move on now to how we were deceived, okay? Because I always say you can find the kitchen sink in there if you go out to enough cycle threshold. You know, if you go way above 40, anything's found in there. And the mathematician that did the analysis did find other things in there. Can you believe it? He found hepatitis, Zika, measles, Marburg, and Ebola, the sequences for all those in the bronchoalveolar lavage fluid of the 41-year-old Chinese man. That just shows you that you could basically find anything in there, including SARS-2.
And for the clarification, perhaps, of our listeners, when you say more than 40, you say more than 40 revolutions?
More than 40 doublings.
Gotcha.
Because remember, 40 doublings gives you 1 trillion copies.
I got you.
Now, here's where the deception comes in. If you use a primer that is not unique to SARS-2, then you've got a real problem. Then you're finding something positive that is not unique to the virus you're trying to discover is whether it's in you or not, is a very clumsy way to say it.
Yeah.
And guess what? One of the primers that was approved by the WHO is a sequence of 18 base pairs that is found on human chromosome number 8. So that means every single person that they tested for SARS-2 that was human, which means all of us, okay, was positive. Because that sequence, that primer sequence, is actually found in the human chromosomes.
Yes.
So they would do the test on you, and it would be positive on everyone. Now you may say, well, why do they want it to be positive? Again, they wanted to create a pandemic and a fright that would cause people to take the vaccine, which I'm going to get to in just a second.
Quick question before we go to our news break. Then this deception,
yes,
was it from the top down? Did doctors, physicians, hospitals know that this was deceiving, or did they practice the deception without knowing it?
The organizers knew that it was a deception, but the average run-of-the-mill doctor doesn't understand.
Got you.
this.
Ladies and gentlemen, thank you so much for joining us this morning. We're looking forward to you continuing as we continue our discussion with Dr. Reed Sheftall today at right after the news break. We'll be right back as we continue that conversation.